Let's record. Okay, we are recording. Let's move this back here.
So are you gonna?
That's what I'm hoping, yeah. But I would still recommend that you record because if it fails. All right, let's see how well that works. Yeah, that's the. Move it further back here. All right. Okay, so we are recording. Let's go over the study guide. This will include stuff we've talked about. It will include things that we haven't talked about. It will include things that you should have been reading in the textbook. So, study guide. And these are all mixed up. Sorry about that. So, first question, first list of things. Be able to label these parts on the heart, on parts of the heart on illustrations for models. Got a model here. Left and right atria. Upper chambers of the atria. Good? You got that? Yep. Okay. So, above the valve, these are the atrioventricular valves. Anything above them is atria. Anything below them is ventricle. Good? All right. The auricles, those are the things right here. They're part, imagine you have a nice, here's a chamber, but you have little extensions that stick out and then flop down like puppy dog. That's exactly what those are. Word auricle means ear, like little puppy dog ears, but they're an extension of the chamber. So everybody good? All right, let's see. AV node, the atrioventricular node. If you remember, when we drew our heart, see if you can get the marker to work this time. When we drew our heart, we had the atria had two different nodes. The one right by the septa that divides the atria and ventricle is the atrioventricular node. That's your backup pacemaker for the heart. The main pacemaker is here. It's your SA node, which is sinoatrial. Are we good? Should have heard that one before. All right, the AV valves we just talked about. They are the valves between the atria and the ventricles. Good, good. All right, the bicuspid valve, also called the mitral. It's right here. The tricuspid, it's right here. All right. So the bicuspid is over on the left side. Tricuspid is on the right. If it helps you remember that, the T in the alphabet is close to the R in the alphabet. The tricuspid is on the right. Good, good. Now, mitral, another name for the bicuspid because people thought it looked like a bishop's mitre, the hat they wore that was two surfaces. Good? All right. The endocardium. Endo, we already said, is the inside layer of the heart. All of this smooth, slippery stuff, both in the atria and the ventricle, that's endocardium, the inside. Very thin, flat, squamous, slippery. All right. Myocardium. Myo means muscle. It's the muscle layer of the heart, the middle layer of the heart. The M should remind you of the word middle. This thick middle right here, that's myocardium around the ventricle, the left ventricle. Super thick. Because it has to send blood all the way up to the head and down to the toes and get it back to the heart. Much thinner, thin as you can get, right atria. See how tiny and thin that is? Because it's only moving blood from the atria to the ventricle. Good? Good. Doesn't need much thickness because it didn't do that much. Purkinje fibers. Fibers that send electrical activity. If we want to be real picky, I could have said the bundle of His and then the Purkinje fibers. I'm just going to treat it like the whole thing is Purkinje fibers. They are fat, wide diameter muscle cells that conduct electrical signals fast. They're what allow you to send the signal to contract down to the tip of the heart, down to the apex, and then up to the papillary muscles before anything in the ventricle contracts. You want the papillary muscles to contract first because they pull on the chordae tendineae and stabilize the valves. Are you with me? Yeah? Okay. Now, visceral layer of the pericardium, that's the same thing as the epicardium. That's the outside layer. This layer on the outside of the heart, it's so nice we named it twice. It's the epicardium, but it's also the visceral, the stuck to the organ layer of the serous membrane that we call the pericardium. That serous sac around the heart is the pericardium. Are we good? Louder?
Which one do you use to start off?
Both. Both. No one both. All right. Foramen means hole. The foramen ovale is an oval hole that is in a fetal heart. It goes through the interatrial septa. It is a hole that lets blood go from the right atria to the left atria, skipping the lungs. Because remember, the baby doesn't get its oxygen from the lungs, it gets its oxygen from mommy through the umbilical cord. Are we good? Yep. All right. After the baby's born, that's supposed to close, and it forms a little scar that we call the fossa ovale. It's that oval spot. Good? All right, papillary muscles. We already talked about that. These are, papilla is finger-like extension. Papillary muscles stick out from the walls of the ventricle, little finger-looking thing. They are attached on one side, they're part of the wall of the heart. On the other side, they're attached to the chordae tendineae. Those are the first things in the ventricle to contract because they pull on the chordae tendineae, pull them tight. That keeps your valves from backwashing and running blood backwards from the atria, from the ventricles into the atria. You don't want that to happen. You want blood that goes into the ventricles to then go out the aorta or out the pulmonary trunk. How do you do that? You stabilize the valves, papillary muscles. Good? All right. Trabecula carne. This rough texture here. Carne means flesh, like carne asada. Okay. Trabecula are crossbars. The name means crossbars of flesh. It's just these lines, these textured lines in the ventricle. We don't have those in the atria. See it? Yeah. And that's a possible test question. All right, chordae tendineae, it means the tendinous cords. Those are the cords here that stick down to the papillary muscle and let it stabilize the valves. What do I mean when I say stabilize? Ventricles are so strong that if you let them push and nothing's happening to the valve, those push the blood up and backwards and back into the atria. But the end of those flaps have the chordae tendineae on them. If the chordae tendineae are not broken, those papillary muscles pull down on them, and the pressure in the ventricle, it builds, but it can't move the valve. So what happens instead? It opens the valve at the bottom of the aorta, the valve at the bottom of the pulmonary trunk. They're both called semilunar valves, and they go, blood goes out there instead of going back into the atria. If that makes sense, y'all. All right. Question 2. Be able to explain the function of the tricuspid, bicuspid, pulmonary semilunar valve, and aortic semilunar valve. First, let's draw on where they are. The tricuspid valve is on the right side. Bicuspid is on the left. Now, the bottom of The other two valves are called semilunar valves, and we have one at the bottom of the pulmonary trunk. That would be here. And we have one at the bottom of the aorta. That would be, yes, I know, it's curves under. We're doing simplified drawing. All right, so semilunar valve here, semilunar valve here. Those together are called the SL valves. This one is the pulmonary semilunar valve. This one is the aortic semilunar valve. Let me show you on the heart what those look like. Right here, this is the pulmonary trunk. It carries blood from the right ventricle to the lungs. And that's the valve that controls the flow into the pulmonary trunk. This is the aorta, right here, this red. And the aortic semilunar valve controls the flow of blood from the big, powerful left ventricle into the aorta. Are we good? Make sense? Yep. All right. So that's where those two are. The tricuspid controls the flow of blood from the atria to the ventricle. If the blood tries to flow back, it stops it. We'll let it happen. The bicuspid or mitral controls the flow of blood from the left atria into the left ventricle. It won't let it flow back. So, what do these valves do? Right atria is where the process begins with the CO2 blood enters the heart to the tricuspid valve to the right ventricle. Pulmonary semilunar valve, pulmonary trunk, pulmonary arteries and lungs. We've just done two of the valves. We have the valve that's between the right atria and ventricle, and that makes sure blood flows from the atria to the ventricle. Can't go back. Then we go into it's not part of the song lyrics, so we go into pulmonary trunk through the pulmonary semilunar valve. That keeps blood from going back into the heart. Good? Keeps it from going back into the ventricle. On the other side, we have blood coming back into the left atria. We have 4 vessels delivering it. They are 4 veins. These veins are special because they are oxygenated veins. They're carrying blood back from the lungs, so that we've made them red on this model because they're full of oxygen. They're the only highly oxygenated veins we've got. Good? Blood goes from the lungs through the 4 veins into the right atrium. Everybody good with that? Then that oxygenated blood goes through the bicuspid or mitral, and then the valve stops the blood from coming back into the atrium. Ventricle builds up pressure until it opens the aortic semilunar valve, and then the blood goes through that valve into the aorta. Good? That valve stops the blood from going back into the ventricle. Yes?
On the side,
Left comes with the left lung, right comes with the right lung. They go behind the heart, they join. They don't get to each other. Are we good? All right. Let's see. So we've got 2 answered. Name all of the septa, all the dividers in the heart. This is easy. You already know that this is an AV valve. It's the valve in the wall, the septa, between the atria and the ventricle. Same here. Tricuspid is a right AV valve. It's through the right atrioventricular septa. Mitral, bicuspid, that is through the left. That's the left AV valve. That is through the left atrioventricular septa. Foramen ovale, that is between the atria, right? Between 2 atria, so that's the interatrial septa. This thick, powerful muscle is part of the interventricular septa, septa between the ventricles. Are we good? Yep. All right. Be able to identify these cardiac vessels. Left and right coronary arteries are the ones that come straight off the aorta. blood leaves the aorta, semilunar valve, sorry, blood leaves the ventricle, goes into the aorta, leaves the left ventricle, goes into the aorta, semilunar valve closes. The aorta is special. It's almost like a pump by itself. It is an elastic artery, so it swells with blood. It's like a balloon. It overinflates and then it tries to return to its normal size. That's important. Elastic arteries like the heart, elastic arteries like the aorta, they take all the pressure from the heart and they reduce that pressure. Instead of releasing all the pressure at once, they swell and return to their normal size. And most of that blood is then pushed out the aorta, down through the aortic arch, the descending aorta, the rest of the body. But some of that blood goes out the right coronary artery, which you can see here under the right auricle. See it right there on the edge? And some of it goes out the left coronary artery. You can see it was cut right there to make this piece we could remove. But the left coronary artery. Blood that goes out the left coronary artery, it goes over here and it then goes down the left anterior descending artery, LAD. Or in anatomy, we usually say the anterior interventricular artery. If you continue, though, there's another branch. Instead of going down that way, the left coronary artery continues over here, and you have the circumflex artery on the left side. If you continued around further, then you'd have the marginal, the left marginal. On the right side, right coronary artery, we come along, and here goes down into the right marginal. Okay, marginal one's right at the edge, so you have to turn to see a little bit. That right continues around to the back, and you have the posterior descending. Are we good? Got it? Those are the vessels I want you to know for the heart. So, be able to identify these cardiac vessels. Left and right coronary arteries, anterior interventricular, great cardiac vein. For the LAD, carries the blood down. The great cardiac vein is the one that brings it back. Good. You send oxygenated blood down there through the interventricular groove to give oxygen to all the muscles. You bring it back to the great cardiac vein, which then goes into the cardiac sinus, which then becomes one of the three vessels returning blood to the atrium. Right atrium. Good? All right. So we got that. Posterior descending marginal. Yeah, we've done this. Okay. Which three vessels bring deoxygenated blood to the right atrium? Superior vena cava, inferior vena cava, and coronary sinus. Everybody good with that? They're right here. Yep. All right. Tell how cardiac muscle differs from skeletal muscle. It's more branching. It's got a lot more mitochondria because it has to work nonstop. Your heart just has to work. And all of the heart muscle cells are connected to each other through intercalated discs. And that's where they exchange ions. That's why we say if one heart muscle cell contracts, all of the heart is going to eventually try to contract. Because ions in the one are going to move into the others. Good? Yep. All right. So we got that. Let's be able to look at an EKG printout and identify the P, the QRS, the T wave. That is stuff that I was hoping you would have been reading already in the textbook. But let's go ahead and draw really quick. Everybody has seen pictures of EKG tracings. So, talking about the heart tracing, where you have a little clip, then you have a giant clip, and you have a slight. They have names. This is a P. This is the Q, R, S. This is the T. That's the. But what's happening here? This does not show the beating of the heart. That's really important. A lot of people get that wrong. What this shows is the electrical activity that tells the heart to beat. All right? Let's see. I want to find a pretty picture. First, I'll tell you, then I'll find a pretty picture. All right, this first little blip, that's the P wave. That is the electrical activity of the atria depolarizing, getting ready to fire. The QRS, much bigger. There's a lot more. That's the electrical activity of the ventricle getting ready to contract. And the electrical activity of the atria now relaxing. T is the activity of the ventricle relaxing. Are we good? Is this making sense to y'all? All right. Let me look for the pretty picture. Let's try around 692, maybe. All right. 691. But I wanted a. Okay, they took the old picture and they broke it up into 3-4 pages. All right, so if you look at 690 through 694, they have pictures. And what I just wrote on the board about atrial polarization, atrial and ventricle, atrial repolarization, ventricle depolarization, RS, and the ventricular depolarization, that is on page 690, bottom of the right column, there's the picture. That's the electrical activity. The mechanical activity, you can see what's happening on the next page on 690, 691. Actually, that's still just showing the electrical activity. Anyway, and then 694, 695 is where it shows physical activity. This is really easy to remember. You have to have a signal to respond to. Does that make sense? So the electrical activity must come before the physical. Yes?
Will they always come out different? Or is
It should always be in that order. Now, in heart attacks, weird things happen. But it should, and the reason why is because blood flows into the atria and from the atria to the ventricle. It does that passively without the atria squeezing at first. And you get a lot of blood, more than half. I think it's in the 75 to 80% of the volume that the ventricles can hold. Flows in passively, flows through the AV valve. Then you top it off. You finish filling the ventricles as the atria squeeze and they shove the rest of the blood in. Anything they have gets shoved in. Does that make sense? Then they relax. And as they relax, the ventricles start to squeeze. And when they do that, the blood is trying to push upward, but it can't because we had the papillary muscles pulled on the chordae tendineae, so these valves can't turn inside and go up the other way. And what happens is as you squeeze, eventually the pressure gets higher in here than the blood pressure in your whole circulatory system. And the semilunar valves, the semilunar valves are going to open. Does that make sense? So to get the blood to flow out of the heart, you have to have more pressure than the pressure of the circulatory system. That's why doctors worry about people who have high blood pressure. Their heart has to work a lot harder. If their blood pressure gets high enough, they might end up having what we call COPD, congestive obstructive pulmonary disease. Basically, you've got a traffic jam of blood molecules, blood cells. And because the pressure is so high, the heart has trouble making enough pressure to open the valves. Does that make sense? Okay, so moving on. Be able to look at the EKG printout, identify the waves. You would agree we can identify the waves now. Be able to describe what electrical activity, depolarization or repolarization, what chambers are involved. You would agree we talked about that. You know where to find it in the book, yes? Not a lot of nod. Okay? We need to do it again? Okay, good. All right, be able to explain why the sounds of Korotkov occur. I'm talking about stuff they listen to stethoscope. When they put a stethoscope to your arm when they're taking blood pressure, they're listening for the sound of blood squirting past blood pressure cuff. In anatomy, we call the blood pressure cuff a sphygmomanometer. But you squeeze that, tighten up, until no blood can flow past it. Then you let off the pressure just a bit. Keep letting it out until you start to hear sound. Let me give you a comparison. I forgot to bring a balloon today. I was going to bring a balloon. Blow up a balloon. You can pull on the end of the balloon, the mouth part, so tight the air can't get out. You agree? And you could just let it be loose. You're not holding it at all, and all the air goes out. But if you blow it up and then you pull and you relax it a little bit, you get to the point that you have that annoying sound. Am I right? What causes that sound is air squirting past the sides. And the pressure of the air is just barely more than the pressure that you're squeezing and pulling with. Does that make sense? So you hear these sounds of Korotkov, these sounds of the blood of the heart beating. You hear them only when the pressure in the blood pressure cuff is lower than a sealed vessel and higher than I just let the blood flow without any restriction. If you seal it, you don't hear anything because nothing's flowing. If the cuff is so slack, so loose, so little pressure, the blood flows no restriction, there's no sound. The sound you hear is in between the two, squeezing it just a little. So when you're putting a stethoscope there and listening, you're watching the needle go down. The minute that you hear the sound of the heart beating, you're not really hearing that. You're hearing the sound of blood squirt, the sound of Korotkov. The minute you hear that, that's your systolic, your high pressure. Remember we said systolic, Superman, up high, diastolic, down low, and invert. Remember that? That's okay. You're going to keep going until you stop hearing it. Spot where you stop hearing it, that pressure is diastolic, down low. Good? All right. Talk about osmotic and hydrostatic pressure. I'm going to draw a blood vessel. Yeah, I know the mark yourself. All right. I've got a blood vessel. Okay. Yeah, it's time, and I can go get a marker. That'll work. All right, I will see you guys in 10 minutes or so. All right, hold on, let me pause that. No, we're good, we're good. You didn't do anything. All right, Zoom recording. Okay, there we go. That's not all right. So now we have markers. We're going to talk about hydrostatic and osmotic pressure. Let's say we got a blood vessel. Capillary say. Its job is to deliver oxygen to the tissue, right? Good. Now, it's important for you to know that not all capillaries are open. If all your capillaries opened all at once, you wouldn't have enough blood to fill them all. Then you'd go into shock. All right. You okay, Doug?
Yeah.
All right. So here's the thing: tissues release tissue signal. Oh no, I'm low on oxygen. When that happens, the pre-capillary sphincter opens, and you send a little bit of blood through, and then the sphincter closes. So capillary beds have sphincters in the front. Pre-capillary sphincter helps the smooth muscle. All right. Now. Yes, but most blood vessels, unlike capillaries, do not run dry without any blood in them. Yes, you're right. We can vasoconstrict and vasodilate. We can. Go ahead.
Because I only have some type of
Not the same thing. We'll talk about that. Venous valves is what we have to prevent and go backwards.
That's not.
Venous valves are not muscular and they're not sphincters. They're just like flaps. So the blood, when say I'm walking, okay? As I walk, my muscles in my leg get shorter and wider, and they squeeze on the vein. When that happens, the blood wants to go up and down. It's like you squeeze a balloon, long balloons. But the blood can't go down because you have flaps of endothelium that they fold out of the way when blood's going up. But when gravity pulls or when they're squeezing tries to push it down, those flaps come together. Those are your venous valves. I'm talking about actual smooth muscle. Okay. So at the heart end of this capillary or capillary bed, you're going to have a pre-capillary sphincter that pulls flow into the capillary. I'm just drawing one capillary. You have a whole. Anyway, the pressure here by the heart is really high. Pressure from the heart we call hydrostatic. It's just fluid pressure. The heart's a pump and it's fluid pressure. But we're going to talk about hydrostatic and osmotic. Here we go. Osmotic and here we go. Osmotic and hydrostatic. Fluid pressure is hydrostatic. It's generated by the pump, by the heart. And that forces it out of the blood vessel, out of the capillaries. Now, capillaries, super thin. We had talked before, I made a table where I had, oh, well, these are the names of the layers in the heart, these are the names of the layers in the blood vessel. Remember? Capillaries are so thin, they only got one layer. They've got the endothelium. Why so thin? Right. Yes, exactly right. It is the place we got to get the oxygen out. That thin wall makes it easy to transfer gases and nutrients. Good. So, thin wall and water pressure forces it out. Heart pressure forces it out. Hydrostatic pressure. But it is water and ions and nutrients and oxygen. It's not the big plasma proteins. It's not the formed elements, the parts of blood like cells. Those things can't fit through the gaps in the wall of the capillary. They stay inside. What that means is that on this end, say we have plasma proteins here. Down here, we're going to have more. And here it's going to be really concentrated down at this end, down at the venous end. Okay, so this is the arterial end. And over here at the venous end, the blood is more concentrated. Why is it more concentrated? Because you kept losing fluid along the whole way. You lose fluid, you lose some more fluid. I'm using a smaller arrow because it's not as much. You lost most of it at the start, right? Does that make sense? Over here, you got a little bit of fluid, even tinier amount that's moving out because there's not much left to lose. You've gotten more and more concentrated. Wow. But there's another pressure that we call osmotic pressure. Okay? The os should remind you of the word osmosis. Remember, osmosis is the movement of water, right? And you probably remember that water wants to dilute things, right? What that means is that as the blood gets more and more concentrated inside, it's more and more attracted to water. So we're shoving water out. As you move in, water wants to come back inside. Down the vessel. Now, at first, when you're at the arterial end, the osmotic pressure is almost nothing. As you get more concentrated, you pull more in. As it comes in, it doesn't just bring water, it brings the carbon dioxide and the nitrogenous waste. That makes sense? So in every capillary, you've got two things that are in balance. The hydrostatic pressure, which is shoving nutrients and oxygen out, and the osmotic, they go out with the water, and this osmotic pressure that is bringing the water back in, but with waste we have to get rid of. Make sense? Now, there's a problem. You'll always push out more than you bring back in. Never get it all back. And that means you should have your ankles get all puffy with edema and stuff. But that doesn't happen because we got a whole nother system that we'll talk about later called the lymphatic system that drains that extra fluid off and puts it back into the circulatory system. Way up in your thorax. Does that make sense? All right. But you'd agree we've explained osmotic pressure. We know how they do fluid exchange, interstitial fluid, right? Between blood and the. Please nod. Is it making sense? Okay. Good. All right. And how does albumin affect these osmotic pressures? You've heard me say before, albumin is a plasma protein. It's big. It can't leave the vessel. Albumin is the main plasma protein. It's the big thing that can't leave. I mean, there are other things that can't leave, but it's the main one. Albumin, one of its primary jobs is creating the osmotic pressure to suck fluid back into the blood, into the capillary at the venous end. Are we good? All right, we got that. There are three types of capillaries. We have continuous capillaries. It's like a pipe. But it's a super thin pipe. It's 1 cell thick, just endothelium. Then we have sinusoidal ones. Imagine a cracked pipe that has big areas. Sinusoidal means kind of S-shaped. There's missing cells. This lets you do massive exchange of substance super easy. It lets big things get rid of.
What's it called?
Sinusoidal, like sinuses. Here, let me write it on the board for you. Okay, so 3 types of capillaries. We have continuous, we have sinusoidal, and the third one we have fenestrated. That means their fenster is the window. There are little openings in them. Think of it as, you know what a strainer or a colander is? You have these things, like say you're making a macaroni, you boil it, but you need to drain it. You pour it into this colander, this strainer, it has all these holes on the sides, so the water flows out, but the macaroni stays. Those little holes are fenestrations. Okay? Have some blood vessels, some capillaries, like the ones in your kidney, glomerulus, that let lots of fluid through. They have lots of windows, but the windows aren't big holes like sinusoids, so it keeps a lot of the stuff behind.
For the sinus one has just hold.
Yeah, yeah. And the fenestrated capillaries are primarily a kidney thing. Like all capillaries can do this. Osmotic, hydrostatic. Fenestrated just allow a lot more stuff. Sinusoidal allow a lot more stuff. Does that make sense? All right, so let's move on. How they affect fluid exchange, capillaries in your system. Got that? How human affects the chronic. Got that. 3 types of capillaries. Got that. How is blood flow through capillaries controlled? By the precapillary sphincter. Not one for each capillary. What you have is a bed of capillaries, and then a shunt goes to the middle of the bed, and the whole thing is controlled. I want to find a pretty picture. Capillary sphincter. The whole thing is controlled by a sphincter or smooth muscle. And here we go. 3 layers, capillary 7, front page 7. Look at page 712. On 712, you can see an artery and a vein, and you can see the fluid. If it's not going through the capillaries, a lot of the capillaries have these sphincters that will flow through them. And if we don't have the sphincter open, the blood just goes through the shunt from the artery to the vein, arterial to the ventricle. If the capillary is, sorry, if the sphincter is open, the blood will flow through the capillary. Does that make sense? Good. So, all right, so we've got that. What are the 3 layers found in most blood vessels? Now, there are different names for them, but I prefer the names that they call tuning. So let's write them down. So layers in blood vessels. Blood vessel layer. We have the one that is touching the blood. It is in intimate contact with the blood. It is closest to the blood. It is the stuff that is lining the lumen space in the middle. We call that layer the tunica, the tunic or layer, intima, meaning it's in intimate contact. It's touching it. Good? Then we have the layer in the middle, which we call the tunica media. Media for middle, but the M should also remind you this is where the muscle is. Almost all blood vessels, capillaries are an exception. Remember, they only have that one inner layer, that tunica intima. But most blood vessels have muscle lining them, smooth muscle, and the tunica media is where it is. So the M should remind you of muscle. But it's not just muscle. It also has connective tissue. The muscles to pull on. And if you are an elastic artery like the aorta, a lot of that connective tissue is elastin, a very stretchy protein. Does that make sense? If you're the other type of artery, we call those conducting arteries. They just carry the blood. They don't stretch much. They have less elastin and more collagen. Good? All right, so, and then the outside layer, the top, like a plastic Pepsi bag to protect it. This is the tunica externa. The outside layer. We good? Yes? No? All right. Be able to explain how oxygen levels affect capillary sphincters. The answer depends on where we're talking about. In almost all the body, when the oxygen level is low, open the sphincter to send blood there to deliver oxygen because the tissues are needing it. Does that make sense? The exception is the lungs, where it works backwards. If an area of the lung is low in oxygen, like say it's damaged, like by smoking or something, that area of the lung is low oxygen. We don't want to send blood there because it's not going to pick up any oxygen. So in the lungs, if we have a low oxygen area, the precapillary sphincter, the arterioles close, try to keep the blood from going there. You won't get the oxygen, or we'll get very little. Does that make sense?
So one part of your body, like,
You'll get less blood. Get a lot less because you'll get enough to keep the cells in the lungs alive, but they're not going to be any good. They're not going to pick up much oxygen. Does that make sense? All right. Okay. How is the relationship in oxygen and vasoconstriction different in heart tissue from other body? Okay. In most body tissues, if you have plenty of oxygen, you vasoconstrict. You say, I don't that much blood, I'm okay, send it somewhere else that needs more. And then if you need some, you vasodilate in that area. Open up the blood vessels so it's easier to send blood full of oxygen. Does that make sense? Now, heart tissue are different in that the coronary artery, remember I told you that the aorta fills with blood and stretches and then it tries to return to normal? As it tries to go back to normal, it tries to force blood back into the heart, but then the semilunar valve snaps shut and the blood in the artery in the aorta, right? Well, at that point, ventricles are relaxing and blood flows from the aorta into coronary vessels and flows around and gives the heart some oxygen. But when the heart cracks, when the ventricles contract, the heart kind of this. And it's like kinking a garden hose, you reduce the flow. Kink those coronary vessels. There's no sphincter, there's no control like that. So you stop the flow. Sense? Keep the flow in the blood in the aorta, so sending it down into the coronary vessel. All right. Be able to explain ABO and RH blood types, descriptive antibodies, and antigens. All right. Have I already played the video for you guys, the Amoeba Sisters video on blood type? No? All right, I'm going to pause this and play a video for you. Promise if you hear me talk anyway. Excuse me, Gene. Okay, so number 12. What are the three layers of most blood vessels? We've talked about the tunicates. Tunica intima, tunica media, and tunica externa. What vessels are missing layers? Capillaries. They only have that inner layer, tunica intima. How is this related to their pressure? Well, capillaries have to have real low pressure because they don't have any muscle around them. They don't have any tough external layer. They're very thin because they have to have very low pressure because they need to exchange gases. Good? All right. 12, conducting arteries and elastic arteries. Conducting arteries just carry the blood through. Elastic ones stretch and expand with the pressure and then try to return to their normal size. How do arteries differ from veins? Here we have a picture of an artery and a vein. Arteries have thick muscle walls. Veins tend to have thin walls, loose, flabby, more space inside, more space in the lumen, but less muscle to control what happens in the lumen. They still can vasoconstrict and vasodilate, but not nearly as good at it as an artery. Does that make sense to y'all? Okay, got that. Which are at higher pressure? Arteries. Arteries are carrying blood away from the heart. They're closer to the heart. So they're closer to the pump. So they're at higher pressure. Good? Yeah, makes sense? All right. How is this reflected in their structure? Well, the ones that are closer to the pump, closer to the heart, have the thicker walls to resist the pressure. Good? All right. Be able to explain the term anastomosis. It means where vessels come together. Well, it means where anything comes together. But we talk about anastomoses in blood vessels because they're places where vessels grow together. This is more common in veins than it is in arteries because lower pressure things can grow together without the risk of squirting blood out when you're forming the connection. So it's not as easily. Does that make sense? All right. What do papillary muscles do? They stabilize the atrioventricular valves by contracting before the rest of the ventricle. Good? The papillary muscles, right here, sticking out from the wall, they stabilize these valves, the AV valves, the tricuspid, bicuspid, by contracting and pulling on the chordae tendineae. that stabilizes the valves, and that happens before the rest of the ventricle contracts. Good? All right. Let's see. How is timing involved in their function? They have to contract before. That means we have to send the signal down the bundle of His, the bundle branches, up the Purkinje fibers, so they get the signal to contract before the rest of the muscles start contracting. Because we have to stabilize the valves and prevent the blood from washing back into the atria. Good? All right. How do venous valves, this is the question Dean was talking about. How do venous valves, respiratory pumping, and skeletal pumping affect the flow of blood towards the heart? When the blood comes off the capillaries, it's pretty low pressure, right? In fact, there's a nice picture of blood pressure this is arterial blood pressure. I wanted the venous pressure too, but 716 shows arterial pressure. And no, it shows both. And you can see that when we get to the veins, the blood is at much lower pressure than it was in the arteries. You see that? Almost so low that it has trouble going against gravity. It's helped by your muscles. That's why if someone sits too much, the doctor's like, oh, go out and walk more. Because your muscle squeezing will squeeze the veins and push the blood up. And then when they relax the muscle and the next step, the blood tries to come back down. But the venous valves come out from the wall and catch the blood so it can't go down. Does that make sense? Louder? No. That happens if, no, you're fine. That happens if you are just sitting still. When your leg falls asleep, you're sitting on the leg, you're squeezing on the vein, making it possible to trip. When you stand up again, all the tissue that was starving for oxygen starts screaming, give me oxygen! So open up all the stinkers and all the blood runs in. Make sense? Okay. It would be real nice if and you like right here. Here's one of those valves. You can see, okay, that's just wood. All right. Here's one of those valves that it goes like that when the blood tries to go back downhill. These flaps of endothelium come with all those cusp valves. Good. Your veins are full of those because the blood has to come down. Right. Because you gotta get it to the heart so we can send it to the lungs so we can get more oxygen and use it. Yeah. So you're 100% correct. And there are two ways that we can pump it. One is the way I talked about before with walking, but the other one is taking a deep breath. There are blood vessels that are trapped in your ribs and your muscles. When you inhale, squeeze those vessels, push the blood back to the heart. Does that make sense? So we call the two methods of pumping respiratory pumping. So the question here, how do venous valves, respiratory pumping, and skeletal pumping affect the flow of blood towards the heart? All three of those work together to get that super low pressure blood. Here, look at how low the pressure is. There's barely any pressure left. If you didn't have those three things happening, you wouldn't have enough pressure to get the blood to go against gravity back to the heart. Make sense? And it's even more important when we start talking about the lymphatic system because they got no pumps. It's all, it's all respiratory pumping and skeletal pumping. Wow, big muscles. Anyway, we got that. What are the formed elements? I'm talking about red blood cells, white blood cells, and platelets, also called thrombocytes. Good? Those are formed elements. How are thrombocytes different from the other ones? Thrombocytes, platelets, are fragments of cells. They're not complete cells. Good?
So then, so
Right, they're fragments that break off whenever we're making blood. We have some cells that break into 20, 30 pieces, whatever, I don't know. And those little pieces are what's called platelets.
then that's like so
Yeah. Yeah. That happens during blood formation. Okay, so where are we? What would an elevated level of where were we? Okay, what is another name for thrombocyte? It's the platelet. Here will list white blood cell types in order from most common to least. Remember, I wrote it on the board last time. Never let monkeys eat bananas. Neutrophils, lymphocytes. Never let monkeys. Monocytes, eosinophils, basophils. Right? Actually, those would be cytotoxic T cells, which would be a type of. I don't remember what the differential count does when you see an elevated basophil. I know with an elevated eosinophil, that is one of the questions there. It attacks parasites. Right, parasites. So, I did miswrite it. Okay, so, I think I miswrote it. Let's take a look and see. It's all that stuff.
I don't know.
What? You're making me paranoid. What's up?
How are you here?
Okay.
okay.
Okay. Here we go. Differential. And so down to interpretation. Last cell. Cynophils, parasitic, allergic reaction, parasitic, basophils, leukemia, hypersensitivity. Like neutrophils would be common with bacteria. All right, let's move on. Yeah, so I need to rewrite that. Okay. And right here, most common to least. Neutrophils, lymphocytes, never let monkeys, monocytes, eosinophils, and what would an elevated level? Yeah, we got that. What are the most common lymphocytes? They're neutrophils. The way you recognize them is they have a really lumpy looking nucleus. There's a picture. Probably this. Body, vascular. No, we don't. So let me draw. I know I don't have good art skills, but work with me here. If you have a cell that has a nucleus that's shaped like this, it's got lumps and tiny little bridges connecting pieces, stuff like that, that's going to be a neutrophil. It will also have tiny little granules, but they're so small they'll be really hard to see. Okay? Now, if you have basophils, the B reminds you of basic. They have these dark staining granules. And so many of them may not be able to see the nucleus because it's stained so dark purple from all the granules. Not black and purple. Don't have purple. Are you with me? Okay. If it stains red, that's an eosinophil. Same type of thing. You have lots and lots and lots of granules stain red. And you can see a little bit of the purple staining of the nucleus underneath it. Here it's hard to tell because purple granules are purple. But here you'll see underneath that. Like, oh, there's a nucleus, but it's hard to see through all granules. Good. All right. If something is a lymphocyte, the L should remind you that it's little. And the nucleus is going to take up almost the entire white blood cell. There's going to be just a little bit of light staining cytoplasm around the nucleus. Does that make sense? And if it is and you see it's smaller, the lymphocyte's smaller than the others. L and lymphocyte remind you it's little. Good? And then, if it's big, especially if it's nucleus big, kind of like that, maybe. Kind of like a kidney bean or a C or a horseshoe. That's a monocyte. M should remind you that it is massive. Good? And that's the one that will grow up to the macrophage. Okay. All right, so we got that. We are on white blood cell types. 19. We're about to do 20. We're actually in 20 by backtrack. The most common leukocyte, most common white blood cell is the neutrophil, the lumpy nucleus, tiny little granules. These three that have granules, I'm talking about the neutrophil, the eosinophil, the basophil, the basophil, and the eosinophil. They all have granules in them, right? They just have tiny, they're hard to see. They all have granules, so they are called granulocytes. So I could ask you, hey, which ones of the white blood cells, which types are granulocytes? That would be the answer. These, the lymphocyte, the monocyte, these would be agranulocytes.
So what is the granule like?
Well, in this case, the eosinophil, those are chemicals they release to destroy granulocytes. They literally come up next to them. They exocytosis here. Does that make sense?
So
Yes. Granules. In that case, yes, they're different ones, different purposes. We learned that stuff later. What we knew is, oh, they're granules. Yes, when it grows up to be a macrophage, it's like a giant Pac-Man. It kills by phagocytizing. So different ones have different methods. Yes.
Can we take a second?
Sure, we can do a second break. Let me put this on pause. And so we have talked, make sure it started. Did it start? Yes, it did. Okay. So we've talked about blood doping before and how people killed their athletes, how coaches screwed up. Remember, old school blood doping was taking out huge amounts of blood and then putting just the thick parts, the cordelmers, back in. And that made the blood very thick. This question is related to that, but it's not directly the same thing. What is peripheral resistance? That's the resistance of the fluid to flow. Okay. There are a couple things that cause it. One is blood viscosity. But the diameter of the vessel matters too. This makes sense if you think about it. I already told you, we don't want to send blood somewhere we vasoconstrict. If we do want to send blood there, we vasodilate. If you make it bigger, it's easier for blood to get. Now, if you don't believe this, it's easy to prove it yourself. I actually thought about doing this and I didn't. I may have to do it like record a video or something, but get a snorkel like you use when you're swimming in the ocean. Get a straw, normal drinking straw. They get one of those little tiny straws they use for stirring coffee. You try taking a full breath on each one. Easy with the big diameter. Bigger the diameter, the easier it is. Snorkel, easy. Through a straw, it takes you a while. Through a coffee stirrers type straw, it might take long enough to get a full lung that you might just pull it out and start breathing because it takes a while. Does that make sense? So diameter affects flow. The bigger the diameter, the more flow. But there's something else. If you are breathing through a snorkel in the ocean, that's about the same diameter as the garden. But you can move a lot of air through a snorkel really fast. You try exhaling really hard through a snorkel, and you can squirt water across the swimming pool. Seen it, right? You try exhaling that hard into a garden hose to squirt a little bit of water out of the garden. It won't happen. Too long. That's exactly right. So what affects the peripheral resistance? That's the resistance once you're away from the heart, going through all the vessels and everything. Diameter affects it, but length affects it. So diameter of the vessel, the length of the vessel, and the thickness of the substance you're pushing through. Does that make sense? Those are the two biggies. And I'm going to try to find the picture, but there's a picture. Basically, if the cross-sectional area is higher, it's less resistant. So let's look for the picture of peripheral resistance. I think, if I remember correctly, the viewer, or my glasses are on. Sorry. There we go. Vascular peripheral resistance, page 714 to 715. That was really close, around 716. All right. So if you look at this, they have, I'm just going to read it. Resistance is opposition to flow and is a measure of the amount of friction blood encounters as it passes through the vessels. It's on page 714, right column, bottom of the page. Because most friction is encountered in the peripheral systemic circulation well away from the heart, we use the term total peripheral resistance. There are three important sources of resistance: blood viscosity, vessel length, and vessel diameter. You already know more about the sources of resistance than you think you do. If you wanted to drink this thick milkshake, would you pick the short, wide straw or the long, narrow straw? Obviously, the short, wide straw. Oh, because you don't want the milkshake? The internal resistance to flow that exists in fluids is called viscosity. It's how thick or sticky the substance is. The greater the viscosity, the less easily molecules slide past one another. Like our milkshake, blood is more viscous than water. because it contains formed elements and plasma proteins that flows more slowly under the same conditions. Blood viscosity is fairly constant. However, conditions such as polycythemia, an excess number of red blood cells, can increase blood viscosity and so resistance increases. On the other hand, if the red cell count is low, as in some anemias, the blood is less viscous and total peripheral resistance declines. Total vessel length. The relationship between total blood vessel length and resistance is straightforward. The longer the vessel, the greater the resistance, just like you said about garden. That's why a shorter straw is easier to drink from. For example, infant blood vessels lengthen as they grow into adulthood, and so the total peripheral resistance and blood pressure happens. Relationship between blood vessel diameter and resistance is also straightforward. The smaller the diameter, the greater the resistance. Here we go. In a tube of a given size, the relative speed and position. This is important. This is worth highlighting. This is the left column at the bottom, page 715. Yeah. And it says the smaller the tube, the greater the friction, because the relative force of the fluid is contacting that tube wall, which is where the resistance varies inversely with the fourth power of the vessel. Yes, that's math, and yes, we're going to do math. It's not hard math, it's easy. This means, for example, that if the radius of the vessel doubles, the resistance drops to 1/16 its original value. Huge difference, right? So radius to the fourth is 2 times 2 times 2 equals 16, and radius and 1/4 of the radius. So for this reason, larger arteries closer to the heart, which do not change dramatically in diameter, contribute little to the total peripheral resistance. Instead, small diameter arterioles, that can enlarge or constrict in response to neural and chemical controls, are major determinants of peripheral resistance. I'm going to stop reading. You can read all that yourself. But does that make sense? Yeah. So, what is peripheral resistance? It is the resistance to flow of blood to peripheral blood vessels. Usually we're talking arterioles and things that can change in size. Things that are small, smaller diameter, what's happening. Polycythemia, blood's too thick, got too many red blood cells. How are they related to older forms of blood doping? In the old days, they would make the blood too thick and it wouldn't pass through the vessel. Build up red blood cell. Make sense? Be able to explain the difference between arteries and veins in terms of oxygen and carbon dioxide. Okay, in most of the body, not all, I'm talking about the difference in the pulmonary and the systemic circuits. We need to start this again. Sorry. Am I recording already? Okay, that's what I wanted to check. Yeah, you're right. It is. It is. All right, so here's the deal. The systemic circuit is the normal blood flow from the heart to the head, to the tip of the fingers, to the tip of the toes, back to the heart. In those, and that's most of the vessels in the body, arteries are high in oxygen and they're delivering blood to the tissues. Veins are bringing the blood back after the tissue took their oxygen out. Veins are bringing waste and carbon dioxide back. In the pulmonary circuit, that's stuff on the right side of the heart. It's the opposite. Pulmonary circuit, let's just do the song again. You're going to hate the song, I'm sorry. Right atria is where the process begins with the CO2 enters the heart. It's filled with CO2. Through the tricuspid valve to the right ventricle, then the pulmonary arteries and lungs. Once the side of the lungs is done, it's carbon dioxide, takes up its oxygen supply, then it's back to the heart. What that means is the veins going back to the heart in the pulmonary system are full of oxygen. Arteries carrying blood away in the pulmonary system are low in oxygen because they haven't hit the lungs yet. And veins, which are after the lungs and go back to the heart, they are these 4 red things, 2 from each lung, full of oxygen. Those are the only high oxygen veins in the adult body. Sense? All right. Let's see. Have oxygen levels affect capillary sphincters? We already said that. If capillaries if it's not the lungs, if the tissues are low in oxygen, we open up the capillary sphincters, send them some blood, send them some oxygen. When we get to the lungs, we flip around and work opposite. Oh. Okay, once your lungs get oxygen and that part of the lungs doesn't have much, don't send the blood there, it won't fill up with oxygen. Vasoconstrict, squeeze that shut. Does that make sense? Okay. Close the list of oxygen vasoconstriction, different in heart, body, I should say no, got that. Be able to explain ABO and RH type descriptive antibodies at the end. Okay. Antibodies are molecules that your body makes as part of its defense. Antibodies look for things that don't belong and signal white blood cells to come and destroy. Antigens are things that can generate an immune response, things that your body thinks don't belong. So when we talk about the ABO blood typing, and she said, oh, well, AB can take any kind of blood, universal receiver. That's because AB doesn't have anything that recognizes A as foreign, doesn't have anything that recognizes B as foreign. So you give it A, give it B, sure. Give it O, yeah, whatever, because there's nothing on it in the ABO system that shows that. Now, oh, yeah, the universal giver, that's not so bad, but it's hard to get.
That's what I'm saying. You can get to everybody.
Right, but see, the thing that's wrong about all these cutesy cartoons is they act like the only two systems we have are ABO and RH. In reality, blood typing is a hell of a lot more complicated in hospital. So, what I'm showing is not showing up on the screen correctly. Okay, blood typing system. Human blood group system. Here we go. So you heard about ABO, right? And you heard about RH. That's the positive, negative. Look how many there actually are. ABO is the number one big thing to worry about. But MNSP, RH, Smith, Kettle, Lewis, Dumpy, Kid, Diego, YT, XC, Science, stuff, blah, 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 blah. 38 different known blood typing systems. So let me tell you how they actually do blood typing in the hospital. All right, we got blood. We got blood we want to get. We got a patient. We got blood we want to get. Check for the 6 or 7 most common things that cause problems. And then after that, we still don't know because there's 38. Take a little drop of the patient. Like, we don't think it's going to. Yeah, exactly. We don't think it's going to clot, but let's make sure. Let's take a drop of blood. Let's take a drop of the blood we want to give. Let's mix it together. Yep, exactly. You don't even need a microscope.
Just put a light to it.
Yeah, you'll see. Yeah, you put a light underneath. You'll see clots forming if it's going to clot. And you'll be like, oh no, get a different one. Right, exactly. Exactly. That means the body would reject. But yeah, so what they do is they test for. Of course, they test for ABO. They test for like the top 6, 7 things, and then they're like, but double check that, cover your butt because you don't want a lawsuit. And then they literally do it the same way they did it in the Civil War. Drop, smear it together. Did that clot? Make sense? So, when they said, they worded that real careful in that cartoon that I showed you guys. And the cells, they're naked. Well, okay, they do have some other proteins on the surface, but none of the ABO. They said those words, and you heard those words, but it wasn't making it clear that, damn it, look how many systems there are, you know. So, yeah, they had the right idea, but, okay. All right, so where were we?
We were on 35. I'm kidding.
I don't think so.
Wait, hold on. We were on 25 and now we're on 26.
Okay.
Okay.
Be able to explain the meaning of the word isovolumetric. Iso means same. When your heart has a chamber, say the ventricles, they're starting to contract, but they haven't reached enough pressure to open the semilunar valves and have blood leave. Now, gases, you squeeze them and they get to a smaller shape, smaller space they take up. Fluids don't do that very much. So isovolumetric means the heart's contracting, but the volume inside staying the same. That continues until the pressure builds up higher than the systemic blood pressure, and then the semilunar is open and the blood pushes out of the heart. Does that make sense? Isovolumetric just means the pressure is increasing, but the volume is staying the same. Good? All right. How many vessels carry blood to the left atria? Not the right.
3, 2, 1. 3. No, 4.
2 from the left side and 2 left one from the right.
Oh.
We good?
You said vessels, I was thinking like on a smaller scale.
Oh, no, but that would be to the right. Let's go through the right. Okay. So obviously we have the superior vena cava, we have the inferior vena cava. But the third one is that the coronary sinus carries blood back into the atrium. Does that make sense?
That's why I thought you were talking more or less on a smaller scale, like the great cardiac.
Yeah. Well, the great cardiac sinus follow it. That just goes into
Right,
becomes
it's coronary.
coronary sinus. Yeah, all of these drain into the coronary sinus. Yeah. All right, but the trick was left atrium. That's why I tried to emphasize. Okay. What are their names? Those are pulmonary veins. It's the only oxygenated vein. All right, Bill, is there a relationship to pulse pressure, systolic pressure, mean arterial pressure? Pulse pressure is the pressure between the systolic and diastolic. Remember, we got the systolic high, the diastolic low. How are we doing with time?
Oh no, my watch is telling me that it's time to stand up.
Okay. All right. Well,
it's just the
okay. Well, systolic, the high, you remember, diastolic, the low.
Yeah.
The distance between those two, let's subtract, that's the pulse pressure. Okay. Obviously, the systolic high, we said that the mean arterial pressure is the average arterial pressure. In other words, take the difference between these two and the amount of time it's been contracting. Let's actually look it up. I think the formula is you divide it, add it, subtracted it, but here it is on 760. Right column near the bottom. And it says mean arterial pressure is diastolic plus pulse pressure divided by. Okay. We're not going to do amounts and half. But if we had a fully set up lab, we'd be. That's it. There. Highlight that here. So 760. Easy to find. It's the one right next to the diagram pressure. Yeah. If you can't remember the 760, of course you can look it up. But if you can't remember, right there, the diagram. Okay. Let's see. How does the appearance of oxyhemoglobin differ from deoxy? Just tell me which one's bright or red. Yep, oxyhemoglobin. If you have the oxygen, for higher, brighter red. Good. What is the function of serous fluid? It lubricates movements and it holds the two sides of the serous membrane together. For the heart, to lubricate the movements is the most important thing. It doesn't hurt when the heart beats. For your intestines, where you have your peritoneal cavity and all, and that's lined in that serous membrane. Movement again is the important thing, so you don't rub it on each other's feet. The lungs, the support thing is what I already showed you, how a little bit of fluid and the two sides stick together. If for some reason you got a little bit of air in there and the two sides weren't sticking, the lung is elastic. The lung is like a blow up a balloon. It takes energy to open the balloon. Then take energy to have the air come out. So it turns the side of the air out, right? Your lung is identical. Diaphragm drops, so muscle contraction, and air flows in. Good. Diaphragm drops, air flows in. But whenever it's time for the lungs to go back to normal, now you can use your intercostal muscles. Force the air out. That's not normally. Normally just relax the diaphragm, and the lungs become stretchy like balloons. They try to return to normal, and they push the air out. But the reason that happens, so you have a lung. Say around that lung, got pleura. One side of the serous membrane is stuck to the surface. What would that be? Visceral or parietal? Yep. Other surface. I mean, obviously, talking about molecule water of serous fluid separated, right? Here's the deal. The reason the lung works is when the diaphragm drops, it pulls on this serous membrane, pulls on the serous fluid, pulls on the other side of the serous membrane, and that pulls the lung down, increasing the volume of the air. If you get air into space, the lung collapses because it's no longer stuck together and the elasticity of the lung is back.
Hey, well, I love you. I got shot. Awesome.
Yeah, no, that totally could have though, because any puncture that I had thought about shooting, the knife wounds that happen to blood, someone stabbed in the lung, they now have a hole that air can get in and fill the pleural space, the pleural cavity, and the lung collapse brings back. Same thing for scuba divers. We're off topic. So talking about that. Remind me later. I'll tell you how to scuba dive. Sorry. Okay. Sorry. All right. So what's the function of serous fluid? It holds the two sides of the serous membrane together. For the names of the layers of the serous membrane, visceral and parietal, together. Be able to explain how the structure of hemoglobin affects the number of O2 molecules it can carry. Easy. Hemoglobin has 4 globulins in it. 2 alpha globulins, 2 beta globulins. Each one in the middle has a heme. You got 4 hemes. Team carry 1 O2. Good? All right. Which vessels are specialized for gas exchange? Capillaries. Why? Because they're really flipping thin. They don't have the extra layers. All right. Be able to look at the EKG trace and explain what electrical and physical activities are happening during it. You already know. I already looked at it because I think it was back around. There we go. It's like 691, 690, up to 694. Good. But it's real easy. Remember, I said the electrical activity has to come before you have to get a signal from track contract?
Yes.
So when you look at that P wave, that's atrial depolarization. That's the atria getting the signal to contract. But they're not contracting. They're going to start their contraction after the start signal. So the contraction will be offset. It won't be like during the P. It might be the end of the P and the interval before the QRS. Does that make sense? We know it's not during the QRS that we have atrial contraction. Part of why the QRS is so tall is it's got two things happening. It has the atria relaxing. Remember that also made a wave, so relaxing, and the ventricle contraction. Does that make sense? All right, the QRS, I just said, you have the relaxing of the atria, the signal, the polarization of the atria of the ventricle. But the contraction is going to be tail into that, and the the T, the ST interval. We call it bottom of the QRS, and then the T wave. Those, that's when you squeeze it. Then, as we're relaxing and depolarizing, that makes sense. Trace the blood flow through the heart. Right atrium, the process begins. See, but there's the heart. You know the song, right? You know where to find the song. What?
Now I know the song.
Sorry.
That's good. I'm running.
Know where the valves are located, what direction blood flows through. We did that already.
The song, right?
Yeah, that would be part of it. Yeah.
Okay.
Remember, do the tricuspid valve? Yes, we talked about it. Okay. But just in general, song or not, know the four valves, know what they control, the direction of the blood. All right. How the leaky ion channels in the nodes create heart contractions. All right, that is the stuff. We were just looking at it. But here's the thing. There we go. On page 67, okay, the pacemaker cells in the heart. The heart, this is like normal resting potential, -70 millivolts where muscle would sit. But you've got leaky channels. They used to say leaky sodium channels. Last I heard, they were saying it's potassium. But either way, or sorry, less leaky potassium. So the idea, though, is you got abnormal cells. See which one. See if they're still saying it's potassium. Okay, pacemaker potential, special properties of the ion channel. See how we have in these cells, hyperpolarization and action closes both closes the potassium channels and opens slow sodium channels. Okay, so they're saying slow sodium. All right, so they went back. But the idea is you don't rest. And like, remember, a normal nerve cell, you wait for the stimulation, tell it, oh, you need to contract, so you need to send an action, and it opens the sodium gates. These sodium gates are always positive threshold. And then it acts like a nerve. Like, whoa, you open the normal gates, the normal sodium gates, sodium rushes in super positive. You get too positive, you open up the potassium gates, and potassium rushes out. And then it takes a little while to set things. Does that make sense? Yeah. So the practical upside is the pacemaker cells in the AP node, the SA node. SA is the primary. Those things will fire on their own, whether you get a signal or not. Now, vagus nerve signals from the vagus nerve can tell you to slow it down, speed it up, actually slow it down, but they will contract regardless. Like if you take a heart out of the body, it'll contract. It's supposed to be sitting there trying to contract. That's what they do in a specific heart, the heart. Yeah, just keep trying to fire. Now I don't know all the details. I should talk to them because I don't see. But I think they probably would put it in a solution that has nutrients and oxygenated, also healthy while you're flying across the But yeah, they will try to fire. Help. Make sense? Now, why does someone get a heart pacemaker? Because they had a heart attack and killed themselves back, they don't back functional heart cells, scars. So you cut off the blood flow. Every time you have a heart attack, heart place it. Start. To place it. Stem cells back in life. I don't know if they've done it. That's what you hear about stuff like that. Instead of replace the heart damage, how to fix the heart. That would be medicine. All right, so how do weak channels create contractions? They get you closer and closer and closer to the threshold. Good. Was albumin the main blood protein, the little red dots? Okay. Where is it found? It's in blood plasma. What does it do to the pH? It helps buffer the pH. If you get to the hydrogens, it'll pick up hydrogens. It's not just osmotic pressure. What does it do to flow of fluid from the interstitial capillaries? That's what we do there with osmotic pressure. It creates osmotic. Good. What does the term hypovolumetric mean? You're not allowed to say, oh shit, that's not the answer. No, that's close. That means you don't splatter. Yes. That's if you're bleeding out and you're going to go unconscious because you don't have enough blood. Your body is going to try to vasoconstrict to keep the central nervous system getting enough oxygen. They'll try to cut off the flow to the places that are bleeding. But if you go into hypovolumetric shock, you just don't have enough blood left. You stop. With me? How does blood vessel diameter affect peripheral resistance? We did that. You got that, right? Okay. What is angina? Chest pain, like heart pain. This is important. This is the reason why they give people calcium channel blockers. Oh, doc, I have heart disease and I have heart pain. Here, take this pill. What is it doing? Remember, normal muscle. Heart's like normal muscle. It's got a sarcolemma, it's got a sarcoplasmic reticulum, it's full of calcium and calcium fractions. You give them calcium channel block dose. Not enough to block all the calcium, enough to block some. You force their heart to beat slow. Now, that's a problem if they have to run from a mugger because now they're dead. But you force their heart to be slow, so it needs less oxygen, so they don't have chest pain, clogged arteries. Because normally, clogged arteries, the heart's working, you can't get enough oxygen, pain. You treat that symptom, problem, the angina, by slowing the heart rate. Does that make sense? You do that with calcium. All good? All right, and now we're done. And I thank you for waiting the whole time, because it looks like it is about time to go.